Okay, so this is going to be a lecture recording on chapter 27. Chapter 27 contains a huge amount of information that you are not responsible for. So this chapter, in chapter 28, I highly recommend that you do not read the textbook. You just really focus on what's on these PowerPoints themselves, because there's like 17 different sections, tons and tons of pages of information, but we are just going to cover the surface of some things, and we're going to completely omit some of the other things from this chapter. So chapter 27 is going to be on the reproductive system. So basically, what is the reproductive system? It doesn't really function continuously like other organ systems, because we're not actually going to activate this system until puberty itself. So male and female reproductive organs are different, but they do share 4 common tasks. So for example, number 1, they're going to form specialized cells for sexual reproduction. These cells that are meant for sexual reproduction are called gametes. Gametes are different depending on whether or not you're male or a female. So if you're male, the gamete is sperm, and if you're a female, your gamete is ova or eggs. The second common task is going to be to bring these gametes together, the one from the male and the one from the female, so our sperm and our ova, and the process of this is going to be sexual intercourse. The third one is going to be to combine the genetic information that's contained within the sperm and the ova, and once they do combine, this is called fertilization. When they actually fuse, what they form is now a zygote, which is also called a fertilized egg. And the zygote is actually the first cell of a new individual, and all other body cells are going to arise from the zygote itself. The fourth task is going to be supporting the development of the fetus, which is called gestation, as well as the birth of the baby, which is called parturition. Okay, moving on to reproductive organs. Our primary sex organs, or gonads, again, are going to be different depending on whether or not you're a male or a female. If you are a male, your gonads are considered testes. If you are a female, your gonads are going to be ovaries. The type of gonad that's actually present during embryonic development is going to determine the fate. So basically, whether or not you have a male pattern of development or a female pattern of development. When we talk about control and development, there are sex hormones, which are a type of steroid hormone. If we're talking about male, the sex hormones are going to be testosterone. If we're talking about females, it's going to be estrogens and progesterone. And they're going to play huge roles in development and the function of these reproductive organs, as well as our sexual behavior and drives. The sex hormones are also going to influence the growth and development of other organs and tissues in the body as well. We also have some accessory reproductive organs in both males and females, and these are things like ducts, glands, and external genitalia. So the next thing we're going to talk about is meiosis, and hopefully this is a review for a lot of you. I'm hoping you've learned this in biology in high school or maybe biology in college. So basically, gamete formation in both sexes is going to involve the process of meiosis. And this is a special type of division, and it's only going to occur in our gonads. So meiosis does have some of the similar stages as mitosis, but mitosis is for our body cells or somatic cells. We're talking about our sex cells. We're talking about meiosis. Pretty much the general process of meiosis is similar in both males and females, but we're going to go into more specifics when we talk about sperm as well as ova later on. So meiosis, again, when we're talking about the process, the whole purpose of it is to reduce the chromosome number in the gametes by half. So we want to take a diploid organism, which would have 46 chromosomes, and reduce the genetic material in half, which means the same thing as saying reducing the chromosome number, so that we get haploid cells. And the whole purpose of getting a haploid cell is that when they come together to form a zygote, that they don't have twice as many chromosomes as the parental cells themselves. Another purpose of meiosis also, as we'll walk through a couple of these different processes, is to increase the genetic variability of gametes. That's unique from mitosis, because mitosis, we want to produce genetically identical cells. When we're talking about meiosis, we want them to all be genetically different. So just a couple terms before we start talking about, again, you just need to know the general process of meiosis. But before we talk about that, a couple of terms. So sister chromatids, we talked about a little bit during lecture that meiosis follows kind of along the same pattern as mitosis, meaning we have to start with interphase. And interphase is the phase where we go in and replicate our DNA. Well, once we go through interphase and we replicate our DNA, the two strands of genetic information that were copied are going to be attached by a centromere. And what we have instead is two chromatids that make up one chromosome. So the sister chromatids basically are going to form after we go through interphase and replicate our DNA, but before meiosis begins. And again, they're joined together by a centromere. Homologous pairs of chromosomes. So originally, our cell had, remember, we have two copies of every chromosome. So we do form homologous pairs of chromosomes, one coming from the mother and one coming from the father, even after replication. When we talk about homologous pairs of chromosomes, the homologous part, why they are similar, is because they carry the same genes that code for the same traits. So for example, eye color. So the one from mom that's paired with the one from dad in this homologous pair, those would each code for the same traits, like hair, eye color, things like that. We also have non-sister chromatids, and non-sister chromatids are the chromatids, one from the mother and one from the father, within a homologous pair. I'm going to show you a picture in a second to kind of help explain what this means. But non-sister chromatids are not identical to each other because, again, one is from the parent, one is paternal, so from the father, and the other one is maternal, so from the mother. But the great thing about having these non-sister chromatids is that they're actually able to exchange DNA with each other, and the process of the exchange of DNA is called crossing over. And when we exchange DNA and we do this crossing over, that's going to be one component that's able to increase the genetic variability, because it's going to combine both the maternal and the paternal genes on the same chromatid. Other things, when we're talking about chromosomal number, we can either have a diploid or a haploid cell. And again, diploid means that you have two copies of each chromosome in the cell. So all of our body cells are diploid. We have two copies of each chromosome. So we have 23 pairs. When we talk about a haploid cell, so like our gametes, the haploid chromosomal number is 23. So we still have 23 chromosomes and we don't have a pair. However, when our gametes fuse, our sperm and our egg, they form a zygote, and that zygote reestablishes our usual diploid chromosomal number that we have in all of our somatic cells. So here's the picture I was talking about, and no, this is not from your textbook, but I like this picture much better. So let's assume that the blue chromosome over here is going to be from dad, and this one is from mom. After interphase and before meiosis begins, we have interphase, and interphase is where we go in and replicate them. So after we replicate our chromosomes during interphase, this is what the chromosomes look like. okay? The chromosomes are attached, each chromosome has a centromere still. So we know even after replication, we still only have 2 chromosomes, like we had 2 chromosomes over here. How you know whether or not you have how many chromosomes you have is based off of the centromere. So again, this little yellow connection dot right here. What we have replicated and what we have doubled is the number of chromatids. So this is a chromatid, and this is a chromatid. This is a chromatid, and this is a chromatid. So after replication, we still have the same number of chromosomes, but we have doubled the number of chromatids that we have. Because over here, 1 chromosome equals 1 chromatid, 1 chromosome equals 1 chromatid. Over here, 1 chromosome equals 2 chromatids, and 1 chromosome equals 2 chromatids. So after replication. Within each one of these chromosomes, so let's look at the paternal one first, each chromatid carries the same exact genes on it. Because they carry the same exact genes, we call them sister chromatids. So same thing if we look at mom. Within this chromosome, each one of the chromatids making up this chromosome carry the exact same genes. This means that they are sister chromatids. However, if we were to compare, let me switch colors. Let's go green. This chromatid with this chromatid, they may or may not carry the same genes, and most likely they don't. Alleles. They may have completely different things on them. Why? One came from mom and one came from dad. Unless mom and dad have the exact same genetic makeup, which they would not have, they would be not carrying the same information. So because they are not carrying the same information, these 2 chromatids are what you call non-sister chromatids. So genetically unique. Okay, moving on. So meiosis, again, we start with interphase and then we go through our typical stages that we have in mitosis. The only difference is that we have 2 rounds. So we still have our prophase, our metaphase, our anaphase, and our telophase, but then we do it again. So if we're talking about mitosis, you had interphase and then you had M phase, which had mitosis in it. If we're talking about meiosis, you have interphase, you have meiosis 1, and then meiosis 2. There is not another interphase between meiosis 1 and meiosis 2. We go directly from meiosis 1 into meiosis 2. Meiosis 1, in a nutshell, is going to give us 2 cells, and then we're going to divide those 2 cells in meiosis 2 to produce 4 cells. So again, we have 2 nuclear divisions, but we only replicate DNA once, and that's because we only have interphase occurring 1 time before meiosis 1. And the whole goal of going through the 2 divisions is so that we can produce 4 haploid daughter cells. You do not need to know the specifics of every single phase, like the nuclear envelope is breaking, the nuclear envelope is reforming, the spindle apparatus is forming, or anything like that. I'm really just focused on a general understanding of what's happening in each phase and which phases we are increasing genetic variability by specific events. So for example, when does crossing over occur? So again, an overview of meiosis, interphase, which is going to happen before meiosis 1 and meiosis 2. That's where our replication is going to occur. And that's where we're going to get our homologous chromosomes. Within each homolog of the homologous pair, we have our sister chromatids. The chromatids within the homologous pair, if we're talking about on separate chromosomes, those would be our non-sister chromatids. We started with a diploid number, so 2 copies of each chromosome. We end with 2 copies of each chromosome. And I know that's confusing because we went through replication. But because they're still attached at a centromere, we still have the same number of chromosomes. During meiosis 1, we're going to separate those homologous pairs of chromosomes. So one cell is going to get the chromosome from dad, one is going to get the chromosome from mom. And then the whole goal of meiosis 2 is now we're going to separate those sister chromatids and put them into individual cells down here. Another thing I want to point out is that once we complete meiosis 1 is when we have officially become haploid cells. So if you look here, we still have one chromosome in each one. So it's not actually meiosis 2 that accomplishes the haploid status, it is meiosis 1. The only point of meiosis 2 is really just to separate those sister chromatids and to put them into individual cells. So meiosis 1, again, we're reducing the chromosome number, going from a diploid cell to a haploid cell during this phase. We do have some very specific and unique events that occur in meiosis 1. One of them occurs during prophase 1 specifically. And a couple things to think about, we do have points within the sister chromatids and non-sister chromatids that we have. So basically our homologous pair that are touching. So we have synapses. And this is where the homologous chromosomes pair up and they form tetrads consisting of 4 chromatids. So imagine again the same homologous pair that we saw with 2 chromosomes made up of 2 chromatids. Basically the synapsis is there's points at which these 2 homologous chromosomes are physically touching together, which is a great thing because where they're physically touching together, they can actually exchange genetic material. So we can exchange genetic material between the maternal and the paternal chromatids. And that exchange of genetic material is what we call crossing over. So crossing over specifically occurs in prophase 1 of meiosis 1, and it's a way for us to increase genetic variability. During metaphase 1, basically, instead of having just regular chromosomes line up at the metaphase plate, instead we have pairs of homologous chromosomes. And these pairs of homologous chromosomes are actually going to line up along the spindle equator randomly, so that either the parental or the maternal homologous chromosome can be given to either side of the equator. So that also is going to increase genetic variability depending on where they line up along the equator is going to determine which cell they get pulled to. So the next step in meiosis 1 is going to be anaphase 1, and that's where our sister chromatids are going to be basically formed. So we're pulling apart sister chromatids that are representing each homolog are going to behave as a unit. So basically the homologous chromosomes, each still composed of 2 joined sister chromatids, they're going to be distributed to 2 ends of the cell. So they're still going to look like this. They're still attached because we're in meiosis 1. So basically the end result of meiosis 1 is to have 2 daughter cells, and we have 2 copies of the one member of each homologous pair. So each of the daughter cells that we formed, each of the 2, is either going to have the paternal chromosome with its 2 chromatids or the maternal chromosome. Or in the event that something goes wrong, it could have neither. We have also formed at this point, at the end of meiosis 1, we've the haploid chromosome number. So we've reduced the amount of DNA within each cell by half. So this is an illustration of what's going on during prophase 1 and prophase, all the events of prophase 1, metaphase 1, anaphase 1, and telophase 1. I didn't even mention telophase 1 in the slide, so I really wouldn't worry about that. It's basically the reverse of prophase 1, what's going on there. Again, what we're focused on in understanding meiosis is specific events that help increase genetic diversity and understanding the terms like homologous chromosomes, sister chromatids, and non-sister chromatids. So just have a general understanding of what's going on in prophase 1, metaphase 1, and anaphase 1, and anything that may be unique to those phases. Okay, meiosis 2, we have the same exact phases, so we're going through the same rounds of PMAT, prophase, metaphase, anaphase, and telophase. It's pretty much the same, except for that we don't have replication of chromosomes before it begins because there's not another round of interphase, and that the cells that undergo meiosis 2, we're starting with haploid cells and not diploid cells. And this is just a picture right here of what's going on. We are basically, the end result is to take our homologous chromosome and separate those sister chromosomes that are making it up and putting them into individual chromosomes themselves. Nothing really special in meiosis 2. Like, again, just understand we're starting with haploid cells, we're ending with haploid cells. What are we separating during anaphase? We're separating homologous chromosomes and pulling apart the sister chromatids and putting those in individual cells. The end result is still a haploid cell, but now we have 4 haploid cells. Okay, so let's compare a little bit just about meiosis and mitosis. A couple important things to think about is that, again, meiosis is going to reduce the chromosomal number by half. If we're talking about mitosis, we keep the chromosomal number the same. Another thing that's unique to meiosis is that we introduce genetic variability. If we're talking about mitosis, we do not do that. We want every single cell to be genetically identical. So the things that can increase genetic diversity that you should be aware of is the random alignment of homologous pairs of chromosomes, which I mentioned previously. That occurs specifically in metaphase 1, not metaphase 2, because again, we're talking about lining up homologous pairs. Also, the crossing over event that occurs specifically in prophase 1, that's going to increase genetic variability because we're actually exchanging pieces of genetic material between the maternal chromatid and the paternal chromatid. And here's just a general overview that probably has way too much information than you need to know, but you can go in and take a look at this if you want to.